Previously, we looked at the various types of hematopoiesis or hemopoiesis arising from the pluripotent hematopoietic stem cell. In today's lesson, we shall be looking at erythropoiesis, which is a type of hematopoiesis. We shall begin this erythropoiesis from the pluripotent hematopoietic stem cell. So I have my pluripotent, so this is my pluripotent hematopoietic stem cell. So pluripotent hematopoietic stem cell. So pluripotent hematopoietic stem cell. Pluripotent hematopoietic stem cell. Now, this particular stem cell actually has a very large nucleus. It's a very large nucleus. So this is my pluripotent hematopoietic stem cell, very large nucleus. So this is just to represent the nucleus. So this is my pluripotent hematopoietic stem cell. And this pluripotent hematopoietic stem cell will undergo differentiation to give rise to the myeloid stem cell. So what I have here, the myeloid stem cell, also known as the colony-forming unit germ, myeloid progenitor. So the myeloid progenitor or stem cell, myeloid progenitor. And this is actually a multipotent stem cell. Also with its nucleus. So we have the myeloid progenitor stem cell. So this is the myeloid progenitor stem cell. Now, we want to produce red blood cell. We are looking at erythropoiesis. So erythropoiesis, which is the production of red blood cell. At this point, this myeloid progenitor cell, stem cell, is actually a multipotent stem cell, which means that it could give rise to several terminally differentiated stem cells. But we want this myeloid progenitor cell to move in the direction of the red blood cells. And so what happens is that this myeloid stem cell will undergo differentiation to give rise to a stem cell. So we'll give rise to so this myeloid stem cell will give rise to a stem cell called the bus-forming unit erythroid. The bus-forming unit erythroid. So this is the bus-forming unit erythroid. Remember that the nucleus is made up of nucleic acids, and because the nucleus is made up of nucleic acid, and if we are staining a cell with a Romanowski dye or Romanowski stain, the basic part of the stain, which is actually positively charged, will be attracted to the nucleic acid, which is negatively charged. Remember that an acid will always interact with a base, and the basic part of the stain is actually blue in color. And so if this nucleic acid, which is found in the nucleus, interact with the base component of the stain, then it will appear quite bluish. Then the space here, which is the cytoplasm, is actually also basophilic. So all these ones are basophilic species. So, but we just leave it that way. So this is the bus-forming unit erythroid. So the bus-forming unit erythroid, as mentioned earlier on, consists of colonies of erythroid lineage. So remember that colony refers to a group of hematopoietic stem cells. So I have pluripotent hematopoietic stem cell that will give rise to the bus-forming unit erythroid. Then the bus-forming unit erythroid will give rise to, so the bus-forming unit erythroid will now give rise to the colony. It will give rise to the colony-forming unit erythroid. So what we have here is the colony-forming unit erythroid. So colony-forming unit erythroid. So colony-forming unit erythroid. Then this colony-forming unit erythroid will undergo further differentiation. So you realize that this myeloid progenitor cell has given birth to the bus-forming unit erythroid. As mentioned in the previous lesson, the bus-forming unit erythroid actually consists of about 200 or greater than 200 erythroblasts as colonies, whilst the colony-forming unit erythroid has about 8,200 erythroblasts when we do a hematopoietic assay, hematopoietic measurement. So this colony-forming unit erythroid will actually undergo further differentiation. Remember that differentiation is an attempt to achieve specialization. So colony-forming unit erythroid will undergo further differentiation to form what is called the proerythroblast. So this will give rise to, so this actually happens to be the first recognizable, this is the first precursor, the first RBC precursor. This is my first RBC precursor cell. So this is the first RBC precursor cell. The cytoplasm here is slightly basophilic. So the first recognizable RBC precursor cell. So here we have here is so this, what we have here is we have the pronomoblast. Pronomoblast. I want to go with the erythroblast. I prefer that one. So proerythroblast, because it speaks of the type of cell. Proerythroblast, which is the same thing as pronomoblast. So from here, we'll be coming out with nomenclatures. So proerythroblast actually is the same thing as pronomoblast. So proerythroblast or pronomoblast. We also have another nomenclature. We call it the rubri nomenclature. Rubri. And this becomes the rubriblast. Rubri. So the rubri nomenclature, this becomes rubriblast. So we have the colony-forming unit erythroid undergoing further differentiation to form proerythroblast, which is also known as, or, so that is the same thing as, which is or pronomoblast or rubriblast. So these nomenclatures, in terms of the naming, they are the same. Then from the pronomoblast stage, you see, in the language of medicine, any word that begins with the prefix pro means before. So if I say proerythroblast, it actually means before erythroblast. Then technically, if we are talking about before erythroblast stage, then the next stage will be we are in the erythroblast stage. So if this stage is the proerythroblast, then the next stage will be in the erythroblast stage, will be in the nomoblast stage. So this will give rise to, so this is another precursor cell. So this is the proerythroblast, but what we have here, so this will be called So we have, so this one goes to here. So we have the pro. So this is the erythroblast stage. This is, so let me name it properly. So this is erythroblast. Erythroblast. So we are in the erythroblast stage. In fact, the erythroblast stage is actually divided into 3 different stages. It's divided into 3. And these 3 different stages we have in the erythroblast is as a result of the differences in the cytoplasmic staining. And so based on the differences in the cytoplasmic staining, the erythroblast stage is actually divided into 3. And so let's look at something here. So this is the erythroblast stage. This pro-erythroblast, which happens to be the first precursor cell in the RBC lineage. This is the first precursor cell in the RBC lineage. This erythroblast, the nucleus, contains genetic materials. And so we have the genes in the erythroblast stage. This is the stage where hemoglobin synthesis actually begins. So hemoglobin synthesis actually begins in the erythroblast. And for us to be able to synthesize hemoglobin, the synthesis of hemoglobin is under the influence of genes or nucleic acids. So there are genes here. So there are genes that are present in the pro-erythroblast stage. And the genes that are present in the pro-erythroblast will have to be activated so that they will begin the synthesis, or the DNA that controls the production of hemoglobin will have to be activated. Now, there is a hormone called EPO, which means erythropoietin. Erythropoietin. This erythropoietin is actually derived from the kidney. So the kidney is the organ that produces erythropoietin. A smaller percentage comes from the liver. So this erythropoietin, as the name implies, erythro, and poietin means a hormone that stimulates. So if poietin means a hormone that stimulates, what is it stimulating? It is stimulating erythrocyte production. Now, this erythropoietin, in fact, this is the hormone that binds to the myeloid progenitor cells and activates the genes of the erythroid lineage. So when it binds to the myeloid progenitor, it activates the genes of the erythroid lineage, and this myeloid progenitor differentiates not towards the granulocyte, not towards the monocyte, not towards the megakaryocyte lineage, but towards the erythroid lineage. So the binding of EPO to the myeloid progenitor cells will trigger the genes in the myeloid progenitor cells to be activated, and this myeloid progenitor cell will undergo differentiation to form the bus forming unit erythroid, which will also undergo further differentiation to form the colony forming unit erythroid. Now, the colony forming unit erythroid will give rise to the first recognizable, or the first precursor in the RBC lineage, and this is called the pro-erythroblast or the pronomoblast. And when it comes to the rubric nomenclature, it is called the rubriblast. Notice that bus forming unit and colony forming unit, or all the stem cells that will be mentioning in this lineage, they are committed to the production of only one type of cell, and that type of cell is called the red blood cell. And that means that these stem cells are unipotent stem cells. They have only one potential. So this pro-erythroblast, which the nucleus contains the DNA or genes that will code for the production of what? Proteins. Remember, in the central dogma of molecular biology, the flow of information is usually from DNA to RNA. And then from RNA, so from DNA to what? RNA, then from RNA to proteins. Remember that our hemoglobin molecules are proteins in nature. And so this flow of information will technically happen. Now, when we take DNA, which is a nucleic acid, RNA is also a nucleic acid. And so the flow of information from DNA to RNA, so you see both of them are nucleic acid. In other words, they speak the same language. They speak the language of nucleic acid. So when the DNA speaks, it is speaking in nucleic acid language, and therefore the RNA will understand the language of DNA. And so the flow of information from DNA to RNA is technically called transcription. Trans means across. We are only passing the information from DNA to RNA, and that is called transcription. We are just transcribing the information. So this is called transcription. However, when the information is flowing from the RNA to proteins, no, RNA, this is a nucleic acid. Protein is not a nucleic acid. So the information that the nucleic acid, the RNAs are carrying, the proteins will not understand the nucleic acid language. And for this reason, the nucleic acid language will have to be translated for the proteins to be able to get the information right. And so the flow of information from the RNA to the protein is called translation. So simply, we are translating the information. So this is translation. And so in the pro-erythroblast stage or the pro-pneumoblast stage, the genes that are there, well, that is the DNA, there is going to be transcription of the transcription of the information. And this implies that we have a lot of RNA, so there will be a lot of RNA in the cytoplasm of the cell. So we have many RNAs in the cytoplasm of the cell. And because we have many RNA in the cytoplasm of the cell, and the nature of RNAs are that RNAs are acid. Listen to the name, ribonucleic acid. It is an acid. And therefore, at this point, when we are staining this erythroblast, because it is an acid, it will have to interact with what? The basic component of the dye or the basic component of the stain. Remember that the basic component of the stain is usually blue, and therefore the RNAs will pick up a bluish color. And because the nucleic acid they show love for the basic component of the dye, we say that the nucleic acids or the cytoplasm is basophilic. Philic means loving. So this erythroblast stage, the early stage of what? Erythroblast, shows a love for the basic part of the dye. So we say it is what? Basophilic erythroblast. The basophilic erythroblast actually is also known as, so we have basophilic erythroblast or basophilic what? Pneumoblast. So we can say basophilic erythroblast or basophilic what? Pneumoblast. And the basophilic erythroblast in the rubric nomenclature, it is still called the rubriblast. So this is actually the early. So this is the first erythroblast. And therefore, the basophilic erythroblast is also called the early pneumoblast, early erythroblast, or that is the rubriblast. So that is it. So we have the cytoplasm appearing bluish. So the cytoplasm, because of the many RNA, when the DNAs or the genes that are in the DNA here are being transcribed, we'll have a lot of RNAs in here, and these RNAs appears what? Bluish. So we have bluish RNA. So the bluish RNA, so bluish, the cytoplasm will appear bluish. Now, from the basophilic erythroblast stage, we move on to the next stage. So we have another stage. In this stage, remember we are flowing, the information is flowing. We have flown the information from the DNA, which is technically in the pro-erythroblast stage, then to the RNA, which is in the erythroblast stage. Now, the information in the RNA is now going to be flown to the proteins. And so, what technically it means is that we are about to synthesize a vast majority of what? The proteins in the erythroblast. So the erythroblast stage, we are going to synthesize the proteins. So once the proteins are synthesized, so we look here, so we are going to synthesize. So this is the nucleus. So the nucleus, bluish, and so we have done transcription, and the transcription resulted in the production of nucleic acid, and these nucleic acid are actually basophilic. Then these RNAs that are here, which have made the cytoplasm basophilic, these RNAs will undergo translation. So there will be translation here, and that will result in the synthesis of proteins. And the proteins in question here are hemoglobin molecules. Hemoglobin. And hemoglobin molecules, which are proteins, are actually, in general, proteins bear. Proteins, in general, proteins are basic in nature. And because proteins are basic in nature, then when we are staining with Romanowski dye, then these proteins, which are basic in nature, will rather be attracted to the acid part of the dye, and the acid part of the dye is eosin, and eosin color is red. So the cytoplasm here will appear red. It will appear red, but we still have some amount of RNA in there. So we have some amount of RNA, and because we still have, so not all the RNAs in the erythroblast, the early erythroblast, is actually translated. So the remaining RNA, so there will be a mixture of what? Hemoglobin proteins and then RNA, because the translation is not complete. So the remaining RNA will be basophilic, and the hemoglobin will also be, the hemoglobins are eosinophilic. So at the end of the day, we have red and then bluish what? Cytoplasm. So different shades of color. So it is not one color, and because so this is an erythroblast. This is still an erythroblast. But this erythroblast, the cytoplasmic staining is that it's having a bluish color and it's also having a pinkish color, and that makes it to have more than one color, and more than one is called poly. So this is called polychromatophilic So this is a polychromatophilic erythroblast. So we have basophilic erythroblast, polychromatophilic erythroblast. Then the next stage after the polychromatophilic erythroblast, so this is called intermediate. So polychromatophilic erythroblast is also called intermediate nomoblast. So this is the intermediate nomoblast. So if we are going by the nomoblast nomenclature, this is called intermediate nomoblast. And if you are going by the Rubri nomenclature, this will be called the pro-rubricide. So we have the rubricides. So pro-rubricides. So this undergo further differentiation, and then we produce still the same nomoblast or erythroblast. So more of the RNAs, the remaining RNAs will be translated. So at the end of the day, almost all the RNAs will be translated at this third stage, and the cytoplasm appears almost entirely red, with just some small shades of what blue color, but so it appears entirely red, and this is called autochromatic. So it now is assuming the actual color of the red cells. Auto, talking about normal. So this is autochromatic erythroblast, or the late normoblast stage. So we have autochromatic erythroblast or the late normoblast was stage, and this is called the rubricide. This is called the rubricide. And so after the autochromatic erythroblast stage, the next stage is called, the next stage actually is the reticulocyte. So what we have here is the reticulocyte stage. So the erythroblast stage are 3. So this becomes the early erythroblast, intermediate erythroblast, late erythroblast. The early erythroblast is basophilic and therefore called basophilic erythroblast. As transcription occurs, we generate RNA, and RNA are acid in nature. And because they are acid, they will love to be attracted to the basic part of the dye, which is actually blue. So at the end of the day, the cytoplasm will appear what? Bluish. So here will appear what? Bluish. And this RNA here will now begin to undergo translation. And when they are translated, we form proteins. And these proteins technically are acidophilic. That is, they love the acid part of the dye because proteins are basic in nature. And the acid part of the dye is actually red. So at the end of the day, we have a polychromatic. Not all the RNAs are being translated. So we still have some amount of the RNA. So we have a mixture of colors. And that will give us polychromatophilic erythroblast. Then by the time we get to the late erythroblast stage, almost all the RNAs have been translated, and we have what is called the autochromatic erythroblast. And the last stage here, which is the last stage in the bone marrow, is the reticulocyte stage. So in the reticulocyte stage, the cytoplasm appears to be completely rare with only some remnants of RNA. So there's something we need to also understand when it comes to the cell lineages. So when it comes to erythropoiesis, there is something we need to understand, and that has to do with the size of, so we need to understand, so we have the size of the cell, number 1, the nucleus size, so size of the cell. It starts as a large cell, and as it is undergoing differentiation to the terminal differentiated cells, the cell size becomes smaller. So there's a decrease in size along the cell differentiation, and the nuclear size also decreases. There is a decrease in the nuclear size. So it gets to the last stage where the nucleus is completely removed out of the cell, because the matured red blood cell do not have a nucleus. Then there is also a shrinkage of the cytoplasm. There is cytoplasmic shrinkage, and then an increase in the cell number. So there is an increase in the cell number. So let me talk about the basophilia. There is also a decrease in basophilia. So there is a decrease in cytoplasmic basophilia, decrease in basophilia. So the affinity for the basic dye decreases as the cell is maturing. So decrease in basophilia in along the maturation, so the basophilia decreases and the eosinophilic properties of the cell rather increases. So you start with an increase in basophilia, and as it's progressing, the basophilia is decreasing and the eosinophilic properties of the cell what increases. The basophilia of the cells is as a result of the RNAs, whilst the eosinophilia properties of the cell is as a result of the presence of the proteins, I mean the hemoglobin molecule. And there is also an increase in the number of what the cells. There is an increase in cell number as it progresses. Now, all these stages is characterized by mitosis. Technically, cell division ceases at the autochromatic erythroblast stage. At all these stages is characterized by cell division or mitosis. Once the cell gets to the autochromatic state, in fact, the nucleus is completely out, it's almost out, it's very, very small. So cell division actually stops in the autochromatic stage. In the reticulocyte stage, what we have is just remnant. We have remnant of what? RNA, and this remnant of RNA will appear red, and that is called polychromato. So that is a polychromatic. So this is a polychromatic cell, a polychromatic reticulocyte. So it is also, because of the RNA, we will see it as a polychromatic cell when viewed under the microscope. Then the last stage here is the matured RBC, and the matured RBC is completely red. There is no remnant of RNA. There is no remnant of RNA in the matured red cell. It's completely red. The cytoplasm is saturated with red hemoglobin, and hemoglobin is eosinophilic, and the eosin part of the dye is actually red. Now, all these stages, right from the pluripotent hematopoietic stem cell up to the reticulocyte stage, all this is happening in the bone marrow. So all these stages is happening within the bone marrow. So the bone marrow. So all here is in the bone marrow. In the bone marrow. Nothing is outside the bone marrow, because we are talking about postnatal or adult hematopoiesis. Everything is happening inside the bone marrow. Now, from the pro-erythroblast stage, so from this stage, right from the pro-erythroblast stage, to the reticulocyte stage, so from the pro-erythroblast stage to reticulocyte stage, technically it spends 4 days. So from the pro-erythroblast stage to the reticulocyte stage is actually 4 days. 4. So it's actually 4 days. So 4 days. So this is the reticulocyte. 1, 2, 3, 4, 5. So it's actually 4 days from the pro-erythroblast stage. So technically, after the pro-erythroblast stage and we are moving to the reticulocyte stage, each of the stages in front after the pro-erythroblast stage approximately 1 day. So that is 4 days. And from the reticulocyte stage to the matured red cell, so the reticulocyte spends part of its life in the bone marrow and part of its life in circulation, I mean the peripheral circulation. So within the bone marrow, the reticulocyte spend 1 to 2 days there. Within the circulation, reticulocyte spend 1 to 2 days there. So in other words, the reticulocyte spend it will take like 1 to 4 days before it can transform into a matured erythrocyte. So this is 1 to 2 days. Technically from here is 4 days. It's 4 days. It's 4 days. So from the pro-erythroblast state to the reticulocyte stage is actually 4 days, and from here, so it's 1 to 2 days, 1 to 2 days. So approximately the whole erythropoiesis, or from the pro-erythroblast stage up to the mature stage, will be averagely 7 to 8 days. So that is the stages involved in erythropoiesis. The hormone erythropoietin is the stimulator of red cell production. It stimulates the myeloid, it stimulates the base forming unit erythroid, it stimulates the colony forming unit erythroid, it also stimulates the pro-erythroblast, and that will move in that direction, produce more of the RBC's precursors. So erythropoietin actually increases the rate of cell divisions and also speed up the rate of maturation of the cell. So basically, this is all the stages involved in erythropoiesis. Now, for this process to occur, we actually need some raw materials, and these raw materials for the process of erythropoiesis to occur, these raw material majorly nutrients. So we need iron. So iron is needed for erythropoiesis to occur. We need folic acid. We need vitamin B12. So we need vitamin B12. We need amino acids, because the globin is made up of amino acids, or the globin is a protein. And so we need amino acids. We need lipids, because we are producing red blood cells. Recall that the phospholipid bilayer, there is a lipid in the membrane. So lipids are also technically also needed. So we have iron, folic acid, vitamin B12, amino acids. Lipids are needed for this process to actually occur successfully. Now, in our subsequent lecture, we will discuss about how these nutrients, iron and folic acid and vitamin B12 in general, the role they play. In fact, iron is required for the hemoglobin synthesis. Folic acid and vitamin B12, they are utilized in the synthesis of DNA. So we use the folic acid and vitamin B12 for the synthesis of DNA. And if these nutrients are actually deficient, what happens is that the synthesis of DNA is going to be impaired. There will be a failure of DNA synthesis, and the end result is that the cells grows larger than normal. It fails to undergo cell division, because remember that the DNA controls cellular division, and because of the deficiency of folic acid and vitamin B12, we are unable to synthesize DNA, and the cell fails to divide, and they grow larger than normal, and that will usually give rise to megaloblastic cells. And if anemia is in there, then that becomes megaloblastic anemia. So if iron is also absent, there is going to be a decrease in the synthesis of the hemoglobin. So iron has to do with hemoglobin. Folic acid and vitamin B12 has to do with the nucleus, the DNA specifically. So basically, that is the erythropoiesis stages. Thank you.